Hello, good morning or good afternoon, everyone, and welcome to this webinar on our CMOS system. My name is Carla Bellak, and I'm the marketing manager here at Multi-Channel Systems. Before we get started, I wanted to point out two things. First of all, we are recording this webinar, and we will send you the link to the recording during the next days. On the right-hand side, you see a Q&A window. Please make use of the window and type your question in there. Only myself can see your question, and I will read it out to our speaker after the presentation. Today's speaker is Dr. Frank Hofmann. He's our application scientist for neuronal applications. He earned his PhD in biology in Tübingen and joined MCS in 2007. Today he will give a presentation of about 30 minutes, and afterwards, as said before, he's available for the Q&A session. I will now hand over to Frank to get started.
So, hello everybody. Thanks for joining us today. The topic is our new CMOS MEA5000 system for extracellular recording and stimulation. As a brief overview, I will first give a bit of a comparison between the classical MEA systems with passive metal electrodes versus the new CMOS system that we are having a look at today. Then I will describe the chips themselves as you see them over here. Then I will go through the recording hardware and finally demonstrate the recording software. The software consists of two tools, the CMOS MEA control for online recording and the analysis software CMOS MEA tools. Of course, both software packages are still under development, are far from being finished, but I will show you what is available at the moment. And finally, I will give you a quick example of what the analysis can look like.
Let me just interrupt quickly because I think the audience cannot see your cursor.
Okay.
Be able to see.
Okay, so we have the CMOS system now. We are used to the classical MEA systems for quite a while. So what are the differences, what are the similarities, and what are the pros and cons of the CMOS system? In the classical MEAs, you have passive metal electrodes. The channel count goes up to 252. There have been experimental systems with up to 1,000 channels, but they were not really user-friendly and not really it wasn't really possible to make a product out of that. The MEAs are transparent, and the sampling rate can go up to 50 kilohertz. They are for recording and stimulation, and there's one contact to the recording equipment per channel. In the CMOS system, things are a bit different. You have active CMOS sensors in comparison to the passive metal electrodes. The channel count can go up to more than 4,000 channels. In the current system, we have 4,225 recording channels. One of the major problems is that the CMOS chips are opaque, so imaging can get a bit difficult. The sampling rate can go up to 25 kilohertz at the moment. They are also for recording and stimulation. And one of the major issues with higher channel counts in passive MEAs was that where to put all that contact pins to go to the recording equipment. And this is solved in the CMOS chip by multiplexing them row-wise. So you have 65 sensors which go to one contact pad, and they are multiplexed. So the difficulties of the CMOS chips are, number one, of course, the problem if you want to combine it with imaging. The chips are opaque, as I already said, so imaging can only be done with upright microscopes, and even there you actually need either a very good microscope or you need to stain your cells to properly see where the cells are in relation to the electrodes. You produce an enormous amount of data, up to 1 terabyte per hour, which is of course good because there's a lot of data to analyze. However, analyzing such huge amounts of data is not very easy. And another issue that one should be aware of, that CMOS chips are of course light sensitive. So the light conditions during recording should be taken care of. It's not pretty difficult just put something on top of the array which keeps the actual chip in the dark, and then you're fine. But you need to be aware of the problem. Then, of course, the advantages of the system, that there's a very high spatial resolution, and you can do recording and stimulation. And also the stimulation spots are really all over the recording area, so you can basically record and stimulate at any given place within your cell culture or within your tissue preparation. And in every experiment, there's a very high information content. You can actually see signals going back and forth through your preparation. But again, that is of course linked to the difficulty to properly analyze your data. So how do the CMOS chips look like? We have, as I said, 4,225 recording sensors, and in between more than 1,000 stimulation sites. You can't stimulate and record from the same sensor, from the same spot, because you can see that down here, that is the complete chip. Down here, that little dark area, that is the actual sensor array. You can see that close up here, that is the actual sensor. Here is the bonding to the substrate. And the actual sensor array area looks like this. These little areas here, these are the sensor spots. And these larger fields here, these are the stimulation sites. The sensor area contains of 65 × 65 grid of sensor chips, of sensor spots, with either 16 micrometer or 32 micrometer pitch center to center. So that means you have either 1 square millimeter or 4 square millimeter recording area, depending on whether we want very high spatial resolution or the still high resolution, spatial resolution of 32 micrometer. You can have either a smaller or a larger recording area. The chip surface is biocompatible. It's a glass-like surface, and in comparison to the classical MEAs, where there is an isolation layer, but there are small holes in the isolation layer where the actual electrodes are underneath. The sensors are underneath the isolation layer, so they are even a bit more durable and easier to clean than the classical MEAs. They are reusable, just like the classical arrays. We don't have really hard data how long you can reuse them, but at the moment we would say that you can use them as long and as often as the classical arrays at least. There are different chamber options available, which I will show you in a second. So the chambers you can put on the chips are either a cell culture optimized chamber with a magnetic lid and a CO2 permeable foil, which you can see down here, consists of the bottom part, which is glued permanently onto the chip, 4 little magnets to keep the lid in place, and in the lid you have that CO2 permeable and water impermeable foil, so that makes a very nice and handy cell culture chamber. Then we also have available this all-round chamber, which can be used for multiple purposes. It's pretty simple. And then there's a perfusion optimized chamber where you have a perfusion in and a perfusion out. And the idea is that you have an optimized flow of liquid over a tissue preparation, basically. The same perfusion equipment that can be used for the classical arrays can also be used for the CMOS array. The recording hardware might look similar to those of you who have already seen our MEA5100 system. There is a head stage, which you see here, which opens and closes and houses the chip. Then it has integrated 3-channel stimulator. It has integrated temperature control. We can go up to 25 kHz sampling rate on all channels simultaneously. On the interface board, which also comes with the system, you have additional analog inputs and a 16 digital bit in and output for TTLs going in and out. We have a 14-bit AD resolution, and it's absolutely mandatory to use an USB 3 interface to get the data to your computer, which brings us to another point. It's necessary to have an SSD drive, a large SSD drive on your computer to be able to store that huge amounts of data because of the high volume of data flowing through from the system to the computer. You need the USB drive and an SSD drive to be able to write in that at that high speed. So all in all, we say that it is mandatory to buy a computer from us, because only then we can make sure that the system really actually works with that computer. So how does the software look like? Of course, with such a complicated and complex system, the software is one of the most important things that comes with the whole thing. We have the CMOS MEA control, which is for online recording. It can do at the moment scheduled recording, so you can say, okay, give me 10 minutes recording every 2 hours or something like this. You have flexible options for visualizing the data, because of course one of the major issues when looking at over 4,000 channels is that it's simply impossible to show raw data from all channels. That simply doesn't work. And it wouldn't help you anyway, because you're simply not able to look at 4,000 channels and actually see what is happening. So what you need is sort of to boil the activity down to a simple color scheme where you can actually spot where the activity happens. Then you have that little region of interest here, which you can move around. And then from within that region of interest, you have a number of channels where you can see the raw data. This can be smaller and larger, but we can see that later on in the demonstration of the software. So there are several options. What you will actually see here, whether it be the actual amplitude or a channel or a spike count or something, but it boils down to that you have a color-coded display of activity. And with that, you do the navigation, so you see where the activity is happening, and then you can zoom in on the raw data. And there's also a monitoring of the computer performance. You see down here what the CPU of your computer is doing, what the hard drive space, how much hard drive space you have left. So that is really important, because when you're running at maximum sampling rate and doing a few minutes even only of recording, you will fill up your hard drive pretty quickly. The CMOS MEA tools is the offline analysis tool. It has a number of spike detection options available. There are multiple filter options. There are frequency filters and spatial filters. Of course, you can export your data. We have export options directly from the software, but we also can read the data format, which is produced by the online tool through the new multi-channel data manager directly into MATLAB, if you prefer to do the analysis in MATLAB or Python or another software. So here again, we have a spatial and time-wise overview of the activity inside the file that you're replaying. And then again, we have that visualization, that graphical visualization of the data. And again, you can zoom in into the raw data, which you see down here. And you can also generate it. So we move on now to a screen share, where I can show you how the software actually looks like. So over here, you can see the two software packages. This is the CMOS MEA control for online recording. This is the CMOS MEA tools for offline analysis. Let's start with the online tool. Over here, I'm in simulation mode now. You have the connected device, and in contrast to a passive MEA, you need to power up the chip. The chip is an active device. You should power it up properly, and you should turn it down properly if you work with it. So now we power up the device. Oh, and we get an error. How nice. And we are crashing. Great. Okay, let's try again. Okay. So I go again to simulation mode, and again I crash. Okay, not that we didn't try that before. Okay, let's see whether it works. Okay, so what needs to be done when you power up your chip is you need to calibrate it. That procedure used to be pretty complicated. However, now there's an automated calibration procedure included, which runs basically all by itself. You just say calibrate, wait for a few minutes, and then you can start recording. So when I start the chip, I should get something here. Okay, so you now get this color-coded scheme here, which at the moment shows you the maximum amplitude. So in a time window that you can select here, at the moment it's a 200 millisecond time window, you get the maximum amplitude for each chip in 200 millisecond bins as a color code. You can also select to show the mean amplitude, or what is usually most useful when you're working with a cell culture is to give you the spike count. So these are artificial signals now, of course, but this will give you now color-coded within 200 millisecond bins the spike count. And you immediately see activity spots within your culture, and then you can move that region of interest to that specific spot and see the corresponding raw data over here. This is the channels within that region of interest, and that is one channel zoomed in. So you can of course also add an additional region of interest, but you will immediately see that the CPU use goes up. The more channels you show in raw data mode, the higher your CPU load will get. Okay, let's remove that again. Then over here you have the recording options. As I said, there are different start and stop options. You can start manually, you can start with a timer, you can start on incoming events. The same goes for stop. So you can set up an automatic recording manual, like something 30 seconds every hour, like that. Down here we have the computer control. You see the CPU use, you see the disk space available, and also which of the instruments that you are using in the software is at the moment producing the most load. So if you see, okay, there's one thing taking up most of the load and running on a critical level here, you can stop that instrument, for example, or remove the number of channels which are displayed. Down here we have the stimulation control. You can have an overlay of the activity that you see here in the background of the stimulation grid, so that you see where you want to stimulate in relation to your activity. There are 3 different stimulation lines available. You can simply select the stimulation sites like this, and the definition of the stimulation pattern is also pretty easy. You simply take these so-called primitives and drag them down here, and the resulting stimulation file looks like that. They can be rather complex, and what you have to keep in mind is that the stimulation principle is also different as they are in comparison to the classical arrays. So what happens when you do a ramp like this, on a CMOS chip is that a positive ramp going up means you induce a negative current. So something like this will mean that you get a negative current pulse as long as this is going up, no current while this is 0, and a positive current while this is going down. So if you want to have a current pulse negative, positive, then what you would have to program is actually this, a simple triangle. So this triangle, voltage triangle, will give you a negative, positive current pulse. This can also be repeated indefinitely and so on and combined into more complex patterns, but that's the general principle. The 3 different stimulation lines can either be started manually, they can be started together, or they can be started on external trigger events. Then over here, as you can see, we have the display of the raw data with the classical functions of zooming in and out, and one channel is always displayed in zoomed in. You can stop that zoom and have a closer look at the data, and then resume the zoom channel while the rest is being continued undisturbed. So that's the actual options that we have now at the online recording tool. There's also the possibility to show the auxiliary channels, the additional analog channels on the interface board, and the digital events coming in. However, it probably will not be possible to add many more analysis functions into the online tool, because really even a very fast computer is running at its limits just by acquiring the data, showing the data, and putting it into the hard drive. So I do not expect that we will see much more functionality on that side of the tool. We will probably get something that will show us triggered activity, of course, when you do stimulation, but I think not too much more. But the CMOS MEA control software for offline analysis, of course, there are many more options because we are not so limited in terms of computing power, because things can be handled in the time that it takes and doesn't have to be online, of course. So here we can open the files generated with the online tool. We get a spatial and time overview of the activity in that file, and then we can do some filtering. We see the filter pipelines down here, and then also there are already implemented a few analysis functions. Let's have a little demonstration. I open one file, and now I can calculate a so-called activity summary. At the moment, everything is focused on spikes recordings. So what I can do is I can detect spikes with a positive and negative threshold, or positive or negative threshold, and the threshold is set for each channel automatically as a manifold of the standard deviation of the noise. So when I do it like this, everything that is 5 times larger than the standard deviation of the noise, either positive or negative, is detected as an event. And 3 milliseconds after each event, there's a dead time. And the display is shown in 10 millisecond bins. So when I do this, I get something like this. Let's show this a bit larger. Okay, so this is a spatial overview of the file that I've just loaded. The dark spots here are the spots where activity has been detected in a color-coded, in a color-code grayscale. And over here, I have a time overview when that activity happened. So it was pretty much quiet until 0.6 seconds into the recording. This was just a 1.2 second file, and it's still several hundreds of megabytes large. I've chosen that small file to be sure that it can be loaded. But if you do extended recordings, or even only a few minutes, you run into several gigabytes easily. So, okay, so within that file of only 1.2 seconds, I have the possibility to choose an area within that file to be loaded into the RAM of the computer and have a closer look at that. So of course I choose that area here where I have most activity, and then I load that selected data, and now I get again the same color-coded data display that you've seen before. And down here I have again the raw data within the region of interest and one zoomed in channel. So now I can review the data, and there are several spots within that culture. That was from a hippocampal culture preparation, where I actually have quite nice spikes. So for example, this here, that blue spot means that I have very negative values within the 200 millisecond bin, and that probably means that there's activity. And when I zoom in, I see that activity. So that was a bit fast to really follow. So you can type in a delay to make the replay slower, even a bit more. So that value that I'm typing in here means add 50 milliseconds after each frame to make the display slower. And now I can wait for the spots showing up. Of course, I can also use a different scaling. Where is it? There's some activities sometimes up here and down there. Let's have a look here. So for example, this is the sort of activity that you can record. As you can see, if you're used to classical MEA recordings, the noise level is much higher. We are roughly recording plus minus 200 microvolts of noise, but the signal size is larger in the same order of magnitude. So the signal to noise ratio is identical, however, the absolute size of signals and noise is larger. So that is what you could usually expect, something like plus minus 200, 300 microvolts noise. But again, then you can see that the spikes that we're recording here are in the millivolt range. So in the end, as I said, you end up with the same signal to noise ratio. So what we also have already implemented is a Spike Explorer. The Spike Explorer can detect spikes in the same way that you've already seen here. So I can show and align the spikes within the area of data that I've just loaded into my my RAM, and now I can look at those spots where spikes were detected and see an overlay plot of the detected events. So if I click one, it's shown over here. And you see an overlay of all detected events, and you see also the time scale or the time where these events were detected down here. You can also do that automatically, perform automatic region of interest creation. So now all electrodes where a certain minimum number of spikes have been detected will be shown over here. There are export functions directly from that window, and you see them, the data over here and the timestamps down there. And something what is pretty nice, you can do a so-called spike-triggered average. So you take one spot of activity. Let's say we take that signal here, and we generate a spike-triggered averaged movie around that spike. Enlarge it a bit and make it a bit slower. Okay, so you see now, down here you see the spike showing up as a dark and then light spot. And here you see where in time this display up here is at the moment. And you see now I can move back and forth. See, you get a dark spot first, gets negative, then a light spot gets positive. And if you're lucky, you can see actually the action potential running through the sensor spot somehow. It doesn't work with that specific spike now, but I've tried a few spikes and I've seen that sometimes you can really see, okay, here here's the cell body, the action potential gets fired, and then you can see it moving along through the cell culture. We have a movie of that on the website where you can have a look. So that is at the moment what the software is capable of doing. What I didn't tell you so far is that there are different filter options available. You can use frequency filters, bandpass, high pass, notch filters, and so on. And also you can use spatial filters to either smoothen the activity or sharpen it. And you can remove artifacts, because there's in each and every chip, there are 1, 2, 3, 4 different sensors which don't work. That's quite normal, that you will see the same thing when you buy a camera, that there's always the one or the other pixel error. That's just unavoidable with CMOS sensors. So the mode of operation using that software is in fact that you select one area within the file, then you optimize your filters, you optimize your recording, your analysis pipeline, and then you process the whole file. Because processing the whole file, putting it through the filters, can take up quite some time. I mean, this is only 1 second of data now, but when you take a 10-second file and you run it through that rather simple filter pipeline with a bandpass and a Gauss filter, for example, it can easily take 20-30 seconds just to go through that filter pipeline. And if you want to do some more sophisticated analysis, like, for example, spike sorting, it can easily take hours to process a file of a few minutes. So if you are thinking about acquiring such a system, be aware that analysis is a really difficult task and it needs a lot of computing power. So that's what I wanted to show you in terms of demonstrating the software. And just to summarize what I've just told you, with the CMOS system you get a very high spatial resolution, you have high content data, and signal tracking across the sensor field is possible. You see just an image. I couldn't show you the movie, unfortunately, because of the software, the software that we are using to do this presentation. So here you see the cell body, and there you see the action potential running along the axon. This is still a screenshot from the movie that is on the website. The volume of data that you produce is really large, and one of the major problems of the technology is that the combination with imaging is rather restricted. So that is it. If any questions came up, I'm happy to answer them now.
Thank you, Frank, for the presentation. Actually, there was a question about the CMOS chips. So what are the diameters of recording and stimulation electrodes?
Oh, beats me. I think they are 8 microns. The recording spots are 8 microns diameter. And the squares for stimulation, don't know, should be roughly double. If you see that image, over here you can see it. So if this is 8 microns, the recording spots, well, they are much larger. And we found that it's usually not effective to use only one of them, at least in retina preparations. I didn't do extended tests in cell culture so far. But one thing that I forgot is that it's actually a nice feature that usually when you use one electrode in a classical MEA for stimulation, that electrode can't be used for recording at the same time, because you saturate that electrode, and at least for a few tens of milliseconds there's no signal. However, when I use, for example, this stimulation area, I can still record on that one, on that sensor spot, and all the other ones around. So in terms of recording directly on the spot of stimulation, the CMOS system is better than the classical MEAs.
Okay. Then we had a question about the chamber, whether it is compatible with regular MEAs too.
So whether you can glue that chamber,
this one?
Yeah, whether you can glue that on the classical MEA, don't know. Probably you could. I don't know whether the sizes are compatible. Never thought about that.
I'm afraid that you won't be able to close the lid, right? Because it's
yeah, probably. So I would say better not. Maybe they are, but I think they are not.
Yeah, they're a bit larger than the normal MEA rings. So.
Yeah. So let's say no, just to be on the safe side.
Yeah. Then there were some questions about the software. So, what is the cost of the software? And yeah.
The software is, we have the same policy as with most of our other software packages. It comes for free. The upgrades are also for free. Of course, it can only be used with our hardware.
Okay. Another question was whether the activity of a single neuron is recorded by several electrodes. So, how many electrodes record the same neuron?
I can't say exactly how many, but I would guess that, of course, depends also from the whether you have a 32 micron or a 16 micron sensor pitch. But I would say at least 9, maybe more. With a 16 micron sensor pitch, I'd say, let's say the cell sits directly on one sensor, you will get all the ones around, definitely, and maybe even the second row around that. That would be my guess.
Okay. Another question more for preparations. So whether the CMOS chips are appropriate for cultured cells, or also suitable for grain slices.
So we have the most experience so far with retina slice preparations, and for that they are really very good. I have some preliminary data from demos with cell culture, which also worked pretty well. I didn't do any brain slice experiments myself so far, and we also have no customer who did that yet. However, there's no principal thing against it. One little difficulty is that the area which we are restricted in a way that we have to cover most of the area around the actual sensor by the chamber. That's why we have that little hole here. We can't make that hole much larger, and we have a bit of a problem to put a slice grid inside that little area. So you need a rather small slice grid. However, if you solve that, there is no reason why it should be used for the one or the other. As I said, I have no personal experience yet how much the what you need to do, what the stimulation strength has to be. However, most of this system is based on the work of Fromhatz in Munich, and he used also slices and organotypic slice cultures on the CMOS arrays, and they work pretty nicely. Especially the organotypic slices work pretty well. But I'm very curious to see how acute slices will behave on the CMOS chip myself.
Okay, thank you. Yeah, and then question about prices. So what's the rough price for the chips and also for the system?
You're better than prices than me. I think it's the system is 68.
Well, yeah, it's just a little bit higher than a huge MEA system.
Yeah.
Yeah. In the range of the MEA system with
the higher channel counts.
Exactly.
And the chips are the same. They are as expensive as the most expensive MEA chips, and they are also reusable. So I think the cost per data point would be roughly the same.
Yeah. Last question was for the computer specifications. You mentioned that that's quite a challenge.
Yeah. So actually, I did run the system on a laptop for demonstration purposes, but that's really a stretch. To actually work with the system, you need a very fast computer, a tower PC with an iCore 7 processor we recommend. Actually, we say it's mandatory that you buy the system from us with computer, because then we know, okay, we need this computer configuration, we can test it and it works. We had some minor issues every now and then with all our later devices which run on USB 3 connections, that even though by specification all USB 3 ports should be identical and should work, not all of them do. So we prefer to deliver the system with a computer where we know that it actually works.
Yes. Okay, I think all questions should have been answered. So thank you for attending today's webinar. We will, well, yeah. It says here any questions have not been answered. I think we covered all of them, but we will stay online for another 5 minutes or so. So if any more questions come up, the Q&A window is still active. So please feel free to answer to ask your question. As promised, we recorded this webinar and we will send you the link. And please note that the recording of all webinars we did are available on our website. And as always, if you have any question, just give us a call or drop us an email. You see the contact information below. Yeah, we're always happy to support. So thank you, Frank, for the presentation. And thank you all for joining us. Yeah, have a nice day.
Bye-bye.
Bye.